The science behind animal growth factors, unlocking nature's secrets to healthier livestock and pets. In recent years, the field of veterinary science has witnessed groundbreaking advancements that are reshaping how we approach the health and productivity of animals. At the forefront of these innovations is the study and application of animal growth factors, biologically active proteins that play crucial roles in tissue repair, development, and overall physiological function. These specialized molecules have emerged as vital tools in both livestock management and companion pet care. By understanding their mechanisms, producers and veterinarians can enhance growth rates, accelerate recovery from injuries, and even extend lifespans through targeted interventions. The implications span across agriculture, zoology, and biotechnology, making this an essential area of exploration for anyone invested in animal welfare and efficiency. What are animal growth factors? Growth factors are signaling molecules produced by various cells within the body. These small protein-like substances initiate cascades of biochemical reactions that regulate cell proliferation, differentiation, and survival. In animals, they serve as critical messengers during developmental stages such as embryogenesis, wound healing, and muscle regeneration. Animal growth factors specifically refer to those naturally occurring in mammals, including humans, cattle, sheep, pigs, and domestic pets. Unlike synthetic hormones often used in traditional farming practices, these bioactive compounds work harmoniously with existing biological systems rather than overriding them. Their ability to stimulate cellular activity without disrupting hormonal balance makes them particularly appealing for long-term usage. Examples include epidermal growth factor, EGF stimulates skin and mucosal surface renewal, fibroblast growth factor, FGF promotes connective tissue formation and angiogenesis, platelet-derived growth factor, PDGF essential for wound healing and bone marrow production, transforming growth factor beta, TGF-beta regulates immune response and fibrosis processes, hepatocyte growth factor, HGH influences liver function and metabolic regulation. Different species exhibit variations in which types dominate under normal conditions but share fundamental pathways governing similar outcomes. This cross-species consistency allows researchers to draw parallels between human medical treatments involving growth factors and applications seen in veterinary medicine today. The role of animal growth factors in developmental biology. Animal growth factors act as master regulators during key phases of organismic development. From conception through maturity, they orchestrate complex interactions among tissues to ensure proper morphological evolution. For instance, during early fetal development, insulin-like growth factor, IGF, plays a pivotal role in directing skeletal system maturation alongside neural crest cell migration patterns. This intricate coordination ensures optimal organ placement while simultaneously establishing foundational vascular networks necessary for postnatal life functions. Such precise control over developmental trajectories underscores why disruptions at any stage could lead to congenital abnormalities unless compensated by external support mechanisms. How IGF influences skeletal system maturation. Insulin-like growth factor IGFI and its receptor complexes interact closely with osteoblasts, the primary builders responsible for new bone synthesis. Through phosphorylation events triggered upon binding sites located along extracellular matrix components, these interactions promote mineralization rates significantly above baseline levels observed prepubertally. Data collected from longitudinal studies conducted across several breeds demonstrate enhanced peak bone mass achieved when IGF concentrations remain consistently elevated during adolescence periods compared to fluctuating profiles typical prior to puberty onset. Mechanisms driving tissue repair. Processes. Tissue damage inevitably occurs due to physical trauma, disease progression, or age-related degeneration. Herein lies another significant contribution made by animal growth factors, facilitating rapid yet controlled restoration efforts throughout affected regions. When inflicted wounds occur, platelet-derived growth factors PDGF activate fibroblasts responsible for synthesizing collagen fibers, an integral component supporting structural integrity post-healing. Moreover, Vascular endothelial growth factor VEG stimulates neovascularization around damaged areas, thereby restoring blood supply essential for nutrient delivery and oxygenation. Combined actions result in accelerated closure times accompanied by reduced scarring incidents, a highly desirable outcome especially relevant within equine rehabilitation programs focused on performance horses undergoing intense training regimens. Bioavailability challenges and delivery systems. Despite their immense therapeutic potential, effective utilization hinges critically upon ensuring adequate bioavailability following administration routes chosen. Oral formulations typically suffer diminished efficacy because digestive enzymes rapidly degrade most protein-based therapeutics before reaching target locations effectively inside gastrointestinal tracts. To circumvent this limitation, alternative methods incorporating transdermal patches infused with microneedle arrays offer promising avenues currently being explored extensively within pharmaceutical research circles globally. These devices utilize microscale channels created via mechanical puncturing techniques, allowing direct permeability across stratum corneum layers where active ingredients subsequently enter circulation efficiently. Applications across different species. Veterinary applications range widely depending largely upon individual needs dictated primarily by species-specific physiology characteristics influencing absorption capacities differently according to anatomical configurations present uniquely amongst varied taxa classifications. Cattle ranchers benefit immensely from employing recombinant bovine somatotropin (RBST) injections designed explicitly towards increasing milk yield outputs sustainably without compromising quality standards maintained traditionally within dairy industries worldwide. Similarly, canine owners seeking improved joint mobility solutions frequently opt for intraarticular injections containing interleukin-1 receptor antagonist IL-1RA administered periodically every few months based upon clinical assessments performed regularly by certified professionals. Ethical considerations surrounding usage practices. As with any biomedical intervention aimed at enhancing natural capabilities beyond baseline thresholds set forth by evolutionary constraints imposed historically upon given organisms, ethical dilemmas inevitably arise concerning permissible limits acceptable ethically speaking regarding enhancement measures applied externally versus internally derived means alone. Organizations dedicated toward promoting sustainable agricultural practices advocate strongly against excessive reliance solely upon exogenous supplementation protocols, potentially leading unintended consequences affecting broader ecosystems indirectly impacted either positively or negatively depending upon implementation strategies employed cautiously throughout entire food chains spanning from farm gates up until consumer tables, ultimately consuming final products generated thereof. Future directions and research frontiers. Ongoing investigations continue exploring novel frontiers related mainly focusing towards stem cell therapy integration coupled seamlessly alongside conventional growth factor applications aiming synergistically amplify regenerative capacities exhibited naturally by living beings irrespective of chronological aging effects normally experienced universally across diverse vertebrate classes encompassing reptiles, amphibians, birds, fish, etc. Preliminary trials indicate encouraging results suggesting potential future deployment scenarios applicable equally well whether addressing acute traumatic incidents requiring immediate surgical reconstruction procedures necessitating extensive soft tissue grafting operations or chronic progressive disorders characterized predominantly by gradual deterioration phenomena observable notably prevalent among geriatric populations suffering debilitating symptoms progressively worsening year after year due primarily attributable causes rooted deep within mitochondrial dysfunction mechanisms underlying cellular senescence phenomena increasingly recognized, now more commonly acknowledged scientifically. Conclusion: the study of animal growth factors represents a convergence of biology, technology, and practical application that continues to evolve rapidly. Understanding their roles provides invaluable insights into improving animal health, longevity, and productivity across multiple domains ranging from commercial farming enterprises down to personal pet ownership experiences. By embracing scientific knowledge responsibly while maintaining awareness surrounding associated ethical considerations, appropriately balanced judiciously wherever possible, whenever feasible, regardless circumstances prevailing momentary situations. Encountered daily routines practiced consistently throughout respective professions, engaged actively participating ongoing dialogues shaping future directions, unfolding continuously ahead awaiting discovery, realization, actualization, transformation. Possibilities forthcoming, horizon brightening steadily evermore.